Assalamu alaikum, everyone. Good morning and welcome to our webinar. My name is Sami and I am part of the Medical Initiatives team at the Hamdan bin Rashid Al Maktoum Foundation for Medical and Educational Sciences. So our initiatives support the healthcare community through CPT accredited workshops and webinars like this one, as well as our health research grant, which hopes to soon fund high impact UAE-based research in its first cycle. We have a lot of plans for the future, inshallah, so please follow us to keep posted. You can find links to our website and social media in the chat. Now, just for a quick housekeeping points, if you have any questions, please post them in the Q&A box at the bottom of your screen. Due to limited time, a few questions will be selected at the end of each talk, and speakers may try to respond to questions by text when they're not speaking. To receive your CME certificate, please fill in the poll that will pop up at the end of your session. Certificates can be downloaded next week from certificates.ha.ae. You will receive an email when they are ready. So please log in using the same email you used to register for the webinar. Finally, a recording of this workshop will be available on the Medical CPD playlist on your YouTube channel, which is the tag at Hamdan Foundation. And today we're delighted to be joined by a team of researchers from the Mohammad bin Zayed University of Artificial Intelligence in Abu Dhabi. And they'll be sharing their expertise on the topic of AI in healthcare, spanning healthcare accessibility, medical imaging, and computational biology. So in the interest of time, I'll hand it over to them directly to introduce themselves and to begin their talks.
Okay, so I hope you can see my slides now.
Yes, we can.
Great. Thank you. Thanks for the confirmation. Hello all, this is Hisham Cholakkal from Mohammad bin Zayed University of AI. So in today's talk, I'll be discussing about our research work at MBZUAI in the area of healthcare AI, with a special focus on improving the healthcare accessibility in the MENA region. So I'm Hisham Cholakkal. I'm assistant professor at MBZUAI. I work in the area of multimodal foundation models and healthcare specialized multimodal LLMs. And I also work developing multimodal LLMs for the wearables and assistive technologies. I would like to thank all my collaborators and also the funding agencies who have generously supported all the research works I'm presenting here today. I also would like to thank my team who have made significant contributions to the projects or the works I'm presenting here today. All right. So we all know that there is a global shortage of experienced healthcare professionals right across the globe, right? And this shortage is actually much more severe even in the MENA region, right? For the reference, we can see that by the year 2030, it's projected that almost we have a shortage of 2.1 million, experienced healthcare professionals. Like, we have a shortage of that big number. And in many countries in the region, we can see that the ratio of the population to the doctors is as low as 8 or even less than 100, which is actually much lower compared to many other parts of the world, right? So this reduced number of experienced professionals actually cause a problem even for those who are already in the field or all those who are already working, right? Because you can see that it's going to increase the work pressure or workload for the medical professionals. So our research is focusing on how we can reduce or the workload of the healthcare experts or healthcare professionals in the field. Just to understand this better, let's try to have a look at how much a patient spend how as patients spend their time while they come to a hospital for the diagnosis. Let's say a patient is coming to a hospital, maybe the first 10-15 minutes they end up spending time on the registration and also taking the vitals and other regular procedures, right? And then after that, the doctor actually spent almost like the expected average is around 13 to 20 minutes they are expected to spend with each patient, right? This can vary depending on the specialities or depending on the hospital and the workload, right? But still, you can see that even within this 15 minutes, the first few minutes are actually spent, like say that almost 20 to 30% of the time is actually spent for information gathering. That means like asking to the patient, like what are to know more about the symptoms, like say if they say they have certain pain, you may be asking like when it started, all those additional information gathering almost takes almost 20 to 30% of the time, right? Then comes the physical examination and also the diagnosis part, with the most crucial part of the the treatment, right? And then even after that crucial part, there are procedures like the documentation and other part of the treatment that's still there, right? And even after meeting the doctor, the patient go home, and there are additional support that's also required, let's say clarifying some of the doubts, or maybe helping them like with the timely reminders on the medicines, and a lot of things are involved in the whole life, the journey of a patient, like in getting treatment, right? So can we optimize at least some of this, or so that the doctor will spend most of their time in the most crucial part, that's like the physical examination, diagnosis, and the other the most crucial part of the treatment, while if you can introduce some kind of assistive tools here in other part of the workflow that effectively can actually reduce the workload, or on the doctor, right? So that's the thought process, or that's the focus of our research here in my team. So that means like between the patient and the clinician, can we have an assistive interface, AI-based assistive interface that can actually gather all the symptoms, the additional, the informations from the patient, and can it actually summarize all those information so that the clinician can actually look at it, and then, while the patient is physically there, they can do ask something that is missing from the AI-based notes or whatever they receive, and do the physical examination and do the remaining part of the treatment in a much faster way, right? So, and also, like, say if a doctor has identified the disease, then they will still again spend a good amount of time in the documenting the diagnosis and taking the approvals, right? Can we also use AI to support that part of the pipeline as well, right? And in all steps, the most important step here, the treatment, there we the my take on this is like we should not use AI in this part of the treatment. Like that should be fully human based. But then can we optimize? Not no, I'm not saying replacement. It's like can we make this part of the workflow and also the post treatment part, post consultation part of the workflow, like with the help of AI, so that it can be speed up, right? So in a way, like in today's talk, I'll be discussing about the foundation models we built, like the healthcare specialized LLM, that's like biomedics, and a few other models we have developed at MBZUAI, and then how we built, the we have built mobile apps and other interfaces to make it available for the patients, right? So I'll be discussing about the solutions, and then I will discuss about the smart glasses we have developed as an assistive device here at MBZUI, and then we'll also discuss or cover about the ongoing projects from my team. On these ongoing projects, we are also looking for clinical collaborators to, for making it, for strengthening those projects. So I will also discuss about those projects. So next, let's start with the first one, that's building of, multimodal LLMs and foundation models, for tailored for the MENA region. Like that means like building the models that has Arabic and English capability. So let's say there is a patient, let's say, who come to you and say that, doctor, I have a abdominal pain, right? So you're not going to give the medical suggestion or prescription or the medicines directly, right? You're going to ask follow-up questions to know more about the symptom. Maybe you'll be asking like when it started, and how long it's been, which part of the body it is, like is it radiating, or so many follow-up questions you will have. And only through the multi-turn conversation between the doctor and the patient, you're going to identify the disease, and then you're going to give the medical prescription, right? So, however, many of the leading medical LLMs these days, unfortunately, are missing this part of the, they are just good as a question answer machine, but they lose this part of ability to have the multi-turn conversation, which is very important for a good quality diagnosis, right? So at MBZUAI, so we have built a model called biomedics, which with a special focus on building enabling the multi-turn conversation capabilities to the chatbot or the healthcare LLMs. And also to serve to the population here in the MENA region, we need the model to be having the Arabic capability, right? So we also introduce the Arabic English bilingual capability to the model. And another important aspect is like, say, sometime the doctors will look at many aspects and they apply their clinical reasoning capabilities to come to a diagnosis or to come to a conclusion. Can we introduce this kind of a reasoning capabilities to the medical model? That's the another research direction we are exploring here. And how we can train these models with limited amount of training data. Because in the healthcare, getting a good quality medical data is always a problem. So we also wanted to kind of, address that part of the problem here. So with this objective from last 3 years now, we are working on building healthcare foundation models with Arabic and English capability. So first we started in the year 2023, we built a model called Biomedics that has, it's basically an Arabic English bilingual medical LLM, large language model that has only text interaction capabilities, right? So, and then later we have extended it to a multi-model LLM that can also understand different medical image scans, like the radiology data, including the X-ray, MRI, and CT. It can interpret the data and answer the user queries related to such data. And then recently we have introduced a model called Medix R1, which is basically supporting the open-ended clinical reasoning in the sense like it's not just before it give you an answer, it apply a reasoning. So it go through several reasoning steps to comes to the conclusion and provide the guidance. And in the medical for the training of Medix R1, we have also used a technique called reinforcement learning to enable training it with a limited amount of data. So you can see that across these models I have mentioned now, our focus is to kind of, evolve from just a simple question answering machine to more of like a model that can interact with the user through the to and fro dialogues and understanding of multimodality, and then also perform the clinical reasoning. So let me show you a bit more details about the Biomedics and Biomedics 2 projects. Here you can see that we have integrated Biomedics with a Telegram bot where actually user can talk to the model in English or Arabic language, and the model actually replay or respond to the user in the same language as they have interacted. Here is an example. You can see.
Mohammed, can you tell me more about when these symptoms started and if there have been any triggers or events that might have contributed to them?
The symptoms started about 3 months ago. I'm not sure of any specific triggers, but I've been experiencing a lot of work-related stress and a few personal setbacks recently.
Thank you for sharing this information, Mohammed.
So whenever the user is typing, it's going to respond in the text format. If this verbal, it will be Arabic. And here is Arabic.
حسناً، بناء على وصفك، من الممكن أن يكون لديك صداع نصفي، ومع ذلك من المهم استبعاد أي أسباب أخرى محتملة. أوصي بتحديد موعد مع طبيب الرعاية الأولية الخاص بك لإجراء.
All right. And now the another model that I mentioned, like the Medixar one, right? So like I said, just to repeat, the model can be trained with a very limited amount of data, like say 50,000 samples, like, the radiology data and the question answer pairs. And it purposefully introduced the clinical reasoning or medical reasoning capabilities into the model, which like, you can see Let me show you a video showcasing the capabilities and highlighting its importance. We have also benchmarked it across different models, including some of the very recent models from the DeepMind, the Google DeepMind, like the Gamma model. It kind of performs much better. So you can see it from the video here.
Imagine a medical AI that doesn't just pick an option. It gives a clear, open-ended clinical answer, grounded in what it sees and reads. That's what we built with Medix R1, an open-source reinforcement learning framework for training medical multimodal models to respond openly and clinically. And the surprising part? We get these gains with only 51K instruction examples. Our 8B model reaches a 68.8% overall average, outperforming models more than 3 times its size, like MedGemma. And our flagship 30B model hits 73.6%, with greater than 95% accuracy on US MLE self-assessments. Even better, our smallest variant is lightweight enough to run on edge devices, like an iPhone. So what's driving it? Medix R1 is a practical end-to-end system, true open-ended generation, interpretable reasoning, and a single-stage RL pipeline that stays efficient. The key is our composite reward. An LLM judge pushes clinical correctness, while an embedding-based signal recognizes valid terminology and phrasing variants, so the model isn't punished for being correct in a different way. That same composite reward also stabilizes training, avoiding the swings you get with single-signal optimization. This isn't limited to one task either. We tested across 17 medical benchmarks, from text-only exams to complex vision-language clinical reasoning, and saw consistent improvements over strong open-source baselines, especially where open-ended clinical answers matter most. The framework works across 16 medical imaging modalities, and we evaluate with a unified 3-stage framework built around reference-guided LLM judging to keep things fair. Medix R1 shows open-ended reinforcement learning in medical AI isn't just possible, it's practical. Check out our project page for the full details, including the model weights, dataset, codebase, and results.
All right. So now let me take you to another interesting problem we have tried to solve. Let's assume that we train these models like biomedics, Medix R1, and this kind of models like with the training data that is available. And then we have, let's say, we have deployed it with the patient interface, right? So we face a problem like, their knowledge base kind of freezes, like, whenever we stop training and put it on the production or deployment, right? Let's say we completed the training of Medix R1 in the year 2025, December, let's say last year, and then just we put it in the production, right? And then if there is a disease outbreak happen, let's say flu or even Ebola kind of issues happen, right? So that real doctors actually they know that from their experience or from the information they get from their surroundings, they can actually adjust their diagnostic pattern or the diagnosis process they follow in the treatment or even in the pre-filtering or procedures, right? So what changes we were trying to analyze, what changes we need to make in the design of medical LLMs to provide a similar ability. So to study this, we have introduced a game-based simulated platform where we have actually 3 types of agents. One is a doctor agent, there is a patient agent, and a measurement agent, right? So these 3 agents, play different roles in a simulated platform. And as you can see here, the patient can go to a doctor and tell their problem, right? And then there will be a multi-turn conversation between the doctor and patients, right? And then we can see that, sometimes the doctor can also suggest some additional labs checkup, right? So then the patient will go to the lab or the measurement agent or the lab agent that could be like, taking blood report or the radiology, right? And then it come back to the doctor with those results, right? And then, in this kind of a simulated environment, we have also introduced a capability like, we can actually replace any of these 3 agents with the real doctors and, patients, so that they can also inject or, the real information they have to the platform, right, in that game environment. So from the simulation, what we have observed is that it's very important to introduce a certain level of memory or experience to the doctors, right? So because if you just blindly deploy an LLM without any kind of, contextual memory, in the sense like, recent visit memory, it's going to fail, So it's very important to introduce such a contextual memory to any of the LLM you deploy for this kind of interface, which is what we observed from this, simulated platform or, environment. Now, let me take you to, the how we, an example where we have taken this progress or the foundation models we built to, build some mobile app that can interact with the patients or, interact with the people, right? So for example, here is a mobile app that's powered by our Medic 7. So here users can actually talk to the model in a seamless way, just like a telephonic conversation, right? And which is more.
Hi, I have a severe headache and I would like some advice from you.
Of course. When did it start? And is it constant or on and off?
Started about 2 days ago and it has been on and off.
Any nausea, sensitivity to light or sound, or vision changes? Also, have you had headaches like this before?
I feel like I have some sensitivity towards light and this has not happened before. This is a new one for me.
Thanks for that. It could be a migraine, sinusitis, or a tension-type headache. Try resting in a quiet, dark room, staying hydrated. If it worsens, returns suddenly, or you develop any new symptoms like weakness, confusion, or fever, seek medical attention promptly.
Thank you for the advice.
Welcome. Feel better soon.
So you can see that here, the different to the Telegram one I was showing, that was a message based. Here it is like a real telephonic conversation, right? That actually to have such a model that talking to you with almost zero latency, you need to make the model really fast, right? That was another challenge we were trying to solve in building such solutions. Now, once a user interact with the model, like either chat-based or an interface, and then the model can actually summarize the key issues of the patient, and it can actually even support the patient by, helping them to book an appointment with the doctor who is probably geographically nearby, in their location, right? So here is a preliminary demo we prepared for the Seha doctors, like, using a small database of Seha doctors we got from internet. And you can see first step, the model summarize the issues or then identify the doctor who is, having a closer specialty and then help in booking the appointment. So these are the few capabilities or or the, the AI models we have built to streamline the interaction between the patient and the doctor before the consultation, including the booking, facilitating the booking of appointment, taking the symptoms, additional details about the symptoms, and creating a clean summary of the symptoms to the doctor so that they can, do the physical examination of the patient and also the ask follow-up questions, what is missing from the summary, right? So now, sorry. Next, I'll talk about our project on, from the other part of it, like say, to related to the wearables and how such wearable devices can help as act as an assistive device for the patient care. So wearable device like smart glasses can actually see what the patient is seeing, right? And then, it can actually listen what the patient is listening to, right? And it can actually also interact with the patient in speech format, right? If you have such a device, you can see what it opens up a lot of opportunities in the patient care, especially as an assistive tool, right? For example, let's say elderly support, or let's say helping a person with difficulty in reading small fonts, maybe helping them in that aspect of it, or even navigating them, like within their home environment, right? So there are a lot of opportunities, or we can that a smart glass or a wearable device can open up, right? And compared to a mobile phone, deploying this kind of an assistive tool on a mobile phone, deploying them on wearables actually opens up opportunity to have this hands-free interaction, which is very crucial in practical, day-to-day life. So at MBZUAI, I'm leading a project called Project Omar, where the objective is to build multimodal LLM or multimodal AI for the smart glasses, and that's also tailored for the MENA region. Like I said, this glass could be adapted to read for the medical prescription, or it could be like even daily care and even the navigation support. Later, I'll also discuss about one of our ongoing project, how this glass could be helpful even in some of the medical documentation. So here we have integrated the smart glass with a robot device actually for supporting the people with limited mobility. So it's kind of a prototype, or it could be also used for the elderly care.
For millions of older adults, simple tasks like reading a prescription or pouring water can be a challenge. With smart glasses, Omer Care captures sound, vision, and gaze. A mobile robotic arm then acts on the user's intent. It empowers people with limited mobility and impaired vision, even making the smallest print readable. At MBZUAI, Assistant Professor of Computer Vision Dr. Hisham Cholakkal is leading its development. Here, he demonstrates by asking the glasses to read a medication label.
Can you tell me what is this tablet?
L-tyrosine, 500 mg.
What is the dose of this tablet? What is the suggested usage?
The dose is 500 mg. The suggested usage is to take one capsule 1 to 3 times daily.
We're reimagining elder care, creating AI that serves humanity.
Now this Arabic capability of the glass.
هل يمكنك أن تخبرني المزيد عن هذا الطبق؟
هذا الطبق هو حمص، وهو طبق شعبي في المطبخ الشرقي، ويعد من الأطباق الرئيسية في الوجبات الخفيفة.
Now, here is another exploratory research we do. Let's say in this work, we used wearable devices that can actually capture the EEG waves. And our objective was to, let's say, check if this signals what you get from the portable EEG device. Can we measure the focus level of the human, like, using some machine learning algorithms? And if you can connect a robot dog with that EEG device, kind of, can we adjust the movement of the dog based on the focus level of the human, right? So this, let me show you a video.
At MBZUAI, researchers have tailored a robot dog with advanced intelligence, giving users the ability to control it with their thoughts. A lightweight brain-computer interface headset picks up brain activity using EEG signals. An AI model interprets those signals into a focus score. When the user concentrates, the robot dog moves forward. This is more than a robotic pet. It's a step toward new human-machine interaction, giving people with limited mobility new ways to gain independence. A glimpse of a future where technology doesn't just respond to what we type or say, it responds to what we think.
All right. So we have discussed about the AI assistants or different assistive tools we have developed at MBZUAI, and have also given a quick overview of some of the assistive device, like wearable devices, including the smart glasses and EEG devices, based works we have. Now let me discuss about some of the ongoing projects in my team. And in fact, I'm looking for clinical collaborators, for all these projects I'm now presenting in the next few slides. So before we deploy any of the solution to the any healthcare setup, we need to have a thorough checking of this, of their performance. Like, we need to validate their performance. So for that, we need to create the benchmarks that are credible and they are closer to the real world usage. So like say, for example, if you want to build, if you want to check how if the mobile devices can be used for improving the healthcare accessibility, we need to build a benchmark for validating it. Or maybe if you want to have a radiology, let's say you have built a best, AI model for radiology report scanning. Before you just go and tell to the radiologist or deploy it for the radiology report generation, you need to validate it, right? But those validations need to be realistic and closer to the clinical usage, right? So we are creating benchmarks for different this kind of scenarios. And another aspect we focus in our research is to build these solutions that are adhering to the regional guidelines, right? And I will also show one of the exploratory research where we show how smart wearables can be used in a medical documentation task and home care. So the first one of the ongoing project, you can see that normally whenever a patient comes, it's not just what the patient say in speech format, or it's not just what you see visually as a symptom, maybe as a, for the skin diseases and all those things, visual symptoms are important. But sometimes even the non-speech audio, for example, if a patient is having a cough or sneeze, right? The audio of that is also very important diagnostic tool, right? Can AI models, or especially the multimodal AI models, understand such cues in, while you actually deploy them? So we are trying to explore this direction. We are building a benchmark that kind of cover different medical cues, including cough and wheezes and all different or non-speech audio cues, along with the visuals like the skin and other visual cues, in a combined benchmark to validate the performance of medical LMs on this task. And another important application, especially may not be that applicable in the UAE, but in other part of the world, some people still will write the things on paper, right? And then the patient have no way to understand what is written by the doctor. So can AI help in understanding it? So we are also exploring that part. So this is one of the benchmark we are building, and we have already built around 5,000 questions in this in this benchmark, but we are looking forwards to expand the scope of the benchmark to make it really comprehensive and useful. And another task is like say radiology report generation. So a benchmark we are creating for the evaluating the VLM's ability for the radiology task. Let's say in the radiology report generation task, it's sometime the physicians or the radiologist actually not just look for the CT or MRI scan what you provide. They may be looking for the EHR, or maybe like say if it's an MRI, they may be also looking into CT and other mode of modality of the scans to come up with a good diagnosis. So can we have this kind of multi-modality information to have a better diagnosis? So we are, and if such information is being provided, whether AI model can leverage it for a better diagnosis. That's one of the research questions we are asking. And another aspect we want to address is that, none of the benchmark are kind of handling the scenario that, let's say, if an AI model or if a doctor itself making a mistake between two closely related, confusing aspects, it is still okay. But then if it is making a totally big blunder, that is like, let's say confusing certain things, which is totally orthogonal, right? So that's a bigger mistake, right? So such mistake need to be penalized higher compared to maybe some ambiguities, or, the making mistakes whenever there is an ambiguity, right? So current benchmarks do not have an option or a way to measure this kind of failure. So we are also trying to build a benchmark that can do that. So we are building. We have. It's been now. This project's been running almost 8 to 9 months now. We have built an annotation platform. The real doctors, we have almost more than 10 medical doctors working on this. They actually look at the scans and identify the, there is a question answer, and they will also see how it need to be evaluated as the rubrics and So we are building it with the help of doctors, this kind of platform. So we really look for collaborations on this project in terms of providing more data if you can, or at least support in terms of more annotations, and so that we can really make it a comprehensive benchmark that can really stress test the VLMs before we deploy, and we are aiming for a publications on top of based on this as well. And the third aspect I would like to bring is the ability of this medical LLM to adapt into the regional medical guidelines. Let's say the models like biomedics or even any other multilingual medical LLMs, they are good at talking the local languages, but then it's not just about the language, right? If you want to deploy it, a model, like say it's not just language in Arabic is enough to deploy it in UAE. You need to know the regional guidelines. Like each country has its own regional guidelines, right? So for example, a BMI of 24 is okay or normal in London, but that's not the case in Mumbai, right? So that's going to be a red flag. So how how good are the medical LLMs for this kind of changing geographical locations? Can they adapt to the regional guidelines? So we are also creating a benchmark to evaluate that, and also we are building a medical LLM that can be better adapted to different regional guidelines and conditions. And finally, I'm also using, or we have also an ongoing project where we are using smart glass for the surgical documentation. Let's say there's a surgeon spent multiple hours to do the surgery, and then after that they spend almost 15-20 minutes to document what they have done, right? This is a very time consuming process. So we are also exploring, if you are giving them a smart glass, we have like the developed in Project Omer, and if the glass knows or have seen the whole surgical procedures, can it automatically prepare an initial draft of the surgery so that the surgeon can just validate it and approve, right? So that kind of simplifies or reduce their workload. So this is another project we are exploring here. So just to summarize what we discussed in today's talk. So we talked about the building of Arabic English bilingual medical LLMs and how they can be helpful in a clinical workflow. Let's say to reduce the help the patients to book an appointment, to also get information gathering so that the doctor can spend their quality time in the physical examination and other important aspect of it. And then we have also discussed about the smart glasses and other wearable devices from we are developing, or the AI for wearable devices we are developing at MBZUAI. And I just would like to conclude by emphasizing that building a solution that is like not just supporting the Arabic language, what we need for the MENA region, what we need is like a solution that can address the local dialects, can refer to the regional guidelines, and so many things to do actually. We just started. It's been 3 years, but there is still a lot to do in this space, and we look for, and I also want to end this talk or discussion with an open call for collaborations to work together to solve this problem, or for building an AI, medical AI that can work in a matured stage for the MENA region. So that's all. Thank you.
Thank
So
you
if you
so
have
much, Professor Hisham. There are many questions actually, so hopefully after your talk, can you please also type some answers out during the next talk? That would be great. But I'll mention a couple now. There's a question by Samar. She asks, how does your model differ from the open evidence besides the Arabic context?
Can you please repeat how the model differs from?
Differ from open evidence besides the Arabic context.
No, the model's been trained with the Arabic data. It's not a proprietary model where we are just doing a prompt, right? So, or not providing a context, right? So because the model being trained with the text that's been translated to Arabic and then being verified by the doctors for their quality of translation and also correctness. So the model, just please don't get confused. I didn't go into the technical details because of the the audience is not technical, right? So that's why. But then it's not a ChatGPT where we are providing Arabic linguistic support. It's more of a model that's been trained with the data that's been curated for with the Arabic language. For example, here in the case of biomedics 2, we have curated with almost like 1.6 million instructions, to train the biomedics 2 data, the model. So yes, it's been curated with the data that has an Arabic language things, and then apart from the linguistic part, you can see the MEDICSA1 model we have developed that is one of the first medical LLM in the world where you can actually do the open-ended clinical reasoning with the help of medical reinforcement learning. So that's another aspect of it as a different or how we are distinct. And the third aspect I was trying to emphasize while we introduce the biomedics is that its ability to interact with the user in a to and fro conversation. Like you can actually have a multi-turn dialogue with the patient to know better about the symptom, like you have seen in the several demos I have shown. Yeah, so these are the three kind of key points I would like to highlight how we are different from the many other players in the market.
Got it. There's a follow-up question to that there was a 2026 Nature Med paper that basically mentions that general LLMs beat specialized clinical AI models. So how do you feel about that, or have you seen
So
that research?
yeah, this is like I have seen that paper as well. These are different studies, and then that paper could be correct, but then it also depends on where you are evaluating it, right? So it's very subjective to the benchmark where you say I can I can evaluate it on, let's say, benchmarks like USMLE, right? So already ChatGPT is getting like close number close to 98 or 99, or the very very high number. But then the issue is that are they really so some of those benchmarks are multiple choice questions, right? And then getting a number, beating those, beating the medical LLMs on those, benchmarks are not really reflecting the practical usability of it, right? So that's why we wanted to have we want to work on creating benchmarks that are closer to the practical deployment, so that then even in that setting, if you see that medical LLMs are failing to be the general purpose LLM, that means that will tell us that there is a problem in the way we are developing the medical LLM, right? So that doesn't mean that we should not develop a medical LLM. That's only saying that, look, there is a mistake or there is a problem in the way we develop it. So we have to relook at it. And obviously, like a specialist is going to be always, or it is expected to be always better than a generalist, right? So if it is not working in that way, there is a fundamental mistake or a problem in the way it's been trained, and that we need to rework.
Great.
I hope
Okay.
yeah.
Yes. We really only have time for one more question, but if you could please, there's plenty of questions in the Q&A. If you could just answer them later. So there's a question by Rowaida. She asks, basically, what's the benefit from the patient's perspective for AI improving efficiency? So not just from the doctor's perspective, but from the patient's perspective.
So very quick answer could be like I don't have to be in a long queue at a hospital for the registration. Like by the time I come there, I'm already with my doctor already know my all issues, and then they and I don't have to be in queue for the registration because it's been done through the app already, right? So it's a kind of make the workflow. If you make the workflow efficient, it also reduces their time, right? The time they spend in the hospital as well.
Great.
And they are going to get a better quality care because the doctor now have 10 times. Doctors are not going to increase the time they spend with the patients because they have so many patients to attend. But now, since you have made the the workload to them efficient, they can now spend on more crucial things, asking more crucial questions to you. That way will also be they will be able to pinpoint your exact pain problems or exact issues, right? That will also lead to better diagnostic outcome.
Great. I think we should end it there.
Mhm.
Thank you so much, Professor Hisham.
Thank you.
Yeah. Professor Aziz.
Hello. Good morning. Can you hear me?
Yes.
Great. Okay. So let me share my screen. Can you see the full screen or the way around?
We can see the full screen.
Okay, great. Okay, so good morning, everyone. My name is Aziz. I am an assistant professor also at MBZUAI. Started not long ago. So most of the work I'm going to present today is going to be my previous work while I was based at in the US at Stanford. So my academic journey is started far up in north of Pakistan in the Karakoram Mountains. And my background is computer science, math, and then decided to do a PhD in bioinformatics or computational biology. Went to Beijing, did a postdoc in Norway, Oslo, and decided to go to the US. I was a staff scientist, a senior staff scientist at Stanford for 5 years. And last year I moved to Abu Dhabi to start my own lab. Okay, so my lab focuses mainly on understanding gene regulation in cancer setting. And this we do by developing open source tools and resources, of course, to analyze, integrate, and interpret these large scale multi-omics data sets and also more diverse data set. By diverse, I mean more multi-ethnic cohorts. And we do that by using more complete reference genomes. I will tell you what a complete reference genome is. And the goal is, of course, to understand gene regulation in health and disease to drive or advance precision medicine. So today I will be telling you two sort of vignettes. About one is about some of the frameworks that we built at Stanford, which help on processing large genomic initiatives at Stanford and produce high level research outputs. And the next one story that I call it understanding whether tumor starts from one cell or multiple. That will be a bit more biology at the end of my talk. So we know that the Human Genome Project has transformed the genomic or the genomic medicine. And it was a 13-year long project started in 1990. We spent $1 billion just to sequence one single reference genome. And after the launch of the reference genome, of course, that you could see that the field of bioinformatics or computational biology started since then and has transformed how we see the future of precision medicine. And fast forward today, we can sequence one genome for less than $1,000. Even some companies now claim to sequence a whole genome for under $200. That's huge progress. And then one big problem with the existing reference genome we use as a baseline, whenever we have any clinical sample comes in, we map back to that baseline or the reference genome, so-called. It has a lot of problems. The one major problem is, of course, it's heavily European-centric. 70% comes from just one single random individual from Buffalo. And when we do clinical genomics, looking for specific mutations or other genomic rearrangements, we have to map our genome, whether it's from Abu Dhabi, Beijing, or from Nigeria, to a reference genome which is heavily European-centric, right? And then it's likely that we lose a lot of meaningful clinical information from there. And part of the reason there has been a lot of push by the community to create these initiatives or the large consortiums to diversify and also to complete the reference genome. By complete, I mean the previous reference genome also had a lot of missing parts, which is around 8%. For example, telomeres and a lot of centromeres were not sequenced. So the T2T reference genome in 2022 fixed that problem by sequencing end-to-end. The reference genome is called T2T reference genome. And then yet the diversity element, of course, still is the problem. So the human plant reference genome was the one that tried to fix this issue by increasing the number of individuals to generate this reference genome. This is more like a graph representation of multiple reference genomes. So the initial draft came out in 2023, where they used 47 individuals across the world to create these reference genomes. And in the current version, I think they have 200 individuals, and then in the next release, they are aiming to do 300 individuals across the world. So initial version, they showed that they reduced the variant discovery error by 34%. That's a lot. And then, of course, now there's a lot of regional and also countrywide reference genomes or pan-reference genomes showing up. One recently came out from the UAE. It used 53 Arab individuals, majority from the UAE. You could see on the left side the map shows that majority of these individuals are from UAE, and rest also they are living in the UAE. And they found 3.5% of sequences were absent from the existing reference genome, including the pan-reference genome, and 9 billion population-specific small variants they found, and around 200,000 structural variants in the Arab pan-reference genome. So, of course, these pan-reference genomes are becoming more and more popular. And speaking of clinical genomics, of course, we're going to use these more and more in the coming days to annotate our data and also make this data available for AI and other algorithms to use and make meaningful predictions, and also, of course, early detection of, for example, cancer at a genomic molecular level. Okay, so these different pan-reference genomes, it's great on one side, but since the reference genome came out, there's been a lot of effort to create these large biobanks, whether using just healthy population or disease-specific cohorts. For example, cancer, we have the TCGA, to create these large cohorts to understand the human development in general, and also the disease mechanisms at molecular level. For example, these different cohorts used different annotations, different versions of reference genomes, and the harmonizing is a huge challenge for these large data sets because there is a lot of compute and storage involved. And then, of course, the growing complexity of cancer that even hardens in terms of reproducibility crisis and so on. So the idea is, of course, how we can fix this by creating, of course, scalable and robust infrastructure to doing these multi-omic analysis end-to-end by taking care of the reference genome and different iterations and also different versions of the tools that we use to process the dataset. So at Stanford, we adopted this platform called Equitable that was built at MSK. This is a very patient-specific platform, which is more genome aware and also versions of different tools aware sort of platform, which does end-to-end bioinformatic analysis. So you could see that on the left side, you could see you have a project or a cohort, and then you have individuals that could be anything with a human, mice, or if you are using any model organism. And then you can create samples at different conditions, whether it's tumor or normal, and maybe there are replicates, and then you do a lot of experiments. The experiment can be anything from a sample, genomics, imaging, and so on. And then you run these different analyses on top. These are version control in terms of which tools are using and also which reference genome you are using. And then it provides a very powerful command line interface where you can take the platform actually the data where data is sitting instead of just getting the data, because these genomic data sets are a lot of, if it's human, of course, it's a lot of data policies and restrictions involved, how we can process and store these data sets. And then it also provides a very powerful browser interface, which stores non-identifiable patient information. And then we have an API where we can interact with both the web and the CLI. And all these tools are version control and highly reproducible and highly scalable. So we use this platform, and of course, then we build around this genomic and epigenomic and transcriptomic suite at Stanford to do point mutations, structural variants, copy number, and RNA and several other assay types in this at scale. So this is just a view of the user interface you can see. And what we did was we ingested a lot of large-scale cohorts. Mainly in this one, we did breast cancer within the platform, and then we reprocessed everything using exactly the same annotations, same reference genome, and exact same version of tools across each and single individual analysis. So we had around 9,000 tumor samples, mainly breast, and we performed around 80,000 analysis. And you could see on the right side, we have a lot of high-level impactful studies came out of this platform. It also supported some of the consortium efforts around the Human Tumor Atlas Network, which I will tell one story later in my talk. And in somehow we had around 20 projects and 6,000 individuals, 9,000 experiments, around 80,000 analyses, and had over 1 petabyte of storage. So one study that came out was led by Casper, where what he did was he deleted T53. So we know that T3 is a guardian of the genome, is a gene which is very important. And if mutation happen in this gene, of course, there's a lot of cancer happen. It's a highly mutated gene in all the cancers as well. So what he did, in an organoid setting, in a normal gastric organoids, he deleted T53 and wait and see what happens into more than 2 years in culture. So you could see that these are 3 different replicas, and you could see in this one, in the wild type on top, in the copy number looking at, you could see this just deployed genome. There is no copy number, applications or deletion, but in the later stages, you could see that the chromosomal abnormalities show up. There's a lot of implications and deletions, and those also happen at specific chromosome arms, right? And also what beautifully he did was he used barcoding to look for specific clones. And you could see that there are 3 R1, R2, and R3. These are 3 replicas. And you can see that there are certain clones, so many clones appear at the early stages, but then there is one just takes over like clonal sweep. So it's like more like poly to monoclonal sort of transition happening here that we see using this clonal dynamics. So I will tell you one story later on in more detail on monoclonality and polyclonality. So this is a wonderful platform that for experiment to understand experimental evolution. So these ones came out in Nature. Then another thing that Katie led using again the platform was understanding the germline, how mutations in the germline, specifically in epitopes, can be used as a way to understand cancer initiation. So she looked at 3,800 breast cancers across 5 cohorts from primary to metastatic settings, and she found like germline variants in the highly expressed genes can influence somatic tumor evolution, and they do by modulating this so-called the germline epitope burden. And it can, if it is increasing at early pre-invasive stage, it's good for you, but if it is increasing at later stages, is not good for you. So the germline epitope burden does this by modulating immunoediting at early stages of tumor development. Another powerful study came out again using the platform where we had a lot of breast cancers. We tried to understand the genomic landscape again for primary, and metastatic across these 1,800 breast cancer whole genome study data sets. Again, we process everything using the same annotations, same tools to do point mutations, copy number, SVs, and also complex amplification across different cohorts. And we found that these complex alterations contribute to replication stress and immune evasion, and they persist throughout tumor evolution. And we also built an AI-based model to predict genomic-based breast cancer subtypes using whole genome RNA and also the clinical data sets. So this is the AI model is a classical ensemble approach to do the subtype prediction for these breast cancers. So the subtype breast cancer prediction method became part of a clinical trial. This is I'm showing a phase 2 clinical trial that became part of, and you could see that we use again the Isabel platform to automate the end-to-end analysis of this trial when a patient goes and sees the oncologist, and then if it is fit, an ER positive patient, and the samples are sent to biotech, and we get both sequence in terms of DNA and RNA, and we ingested that in the platform end to end, and we run our IC cluster, we call it any cluster, and then based on the subtype, the patient can be in a specific cohort in terms of the treatment, and we then also we do sequencing for after the treatment as well, and that also gets processed through the platform. So before I left, we had around 25 patients that been through the pipeline, and the turnaround was 8 to 10 hours end to end. That's significant. Before the platform we integrated, it was taking 2 to 3 days for the analysis and so on. So this is like a sort of one-click analysis where you can get the final reports for the subtype of these patients. So currently we are expanding this large genome study. The previous one I showed was mainly European cohorts. Now we are expanding to doing it multi-ethnically. For example, we have a cohort from Nigeria. We have a cohort also from Malaysia, South Korea, and I'm also building one. We are building one in Pakistan at the Shaukat Khanam Cancer Hospital, and likely one in UAE. And if there are clinicians in this session want to collaborate on this, I'm pretty open to talk more. And so this is happening in collaboration with Stanford Medicine. So we also have one cohort from Mexico, and of course we have a lot of European cohorts. Okay, so this was just the very high level walk you through a couple of studies that came out by using this framework or the platform that we built. And this platform, of course, now in production here at MBZUAI for my lab to use and to process, and then local genomic data at scale and do meaningful studies for local populations and beyond. So one study I will tell you now more in biology or more in science level is it's to understand polyclonal origins of human cancer, whether cancer starts with a single cell or multiple cell. That's been a long debate that we try to understand in this more in colon. So we were part of the Human Tumor Atlas Network, which is called HTAN. This is a Cancer Moonshot Initiative to understand cancer initiation and evolution. Started in 2018 with the aim to create these 3D atlases at cellular, structural, and also molecular level. And you could see that this is a US map, and there's universities across the US they were part of. At Stanford, we were doing both the breast and colon cancer. So we had in total there were 21 tumor sites, 14 atlases, and so on. So I'm going to talk about the colon part, so which I co-led. So we use FAP, which is called familial adenomatous polyposis. This is a condition, a genetic condition. If you have a mutation in a gene called APC, then you develop these polyps in your colon. You could see that this could be very early stage to a later stage like dysplastic, and also some can become malignant and be cancerous. So these polyps can be from 100 to 1,000 at different stages of progression, and patient needs total colectomy by their mid 30s. If these are untreated, of course, they develop cancer and so on. So people with these FAP, the new FAP, they had germline APC mutation. They came to Stanford, and we took their colons out, And you could see that this is a real colon on the table, and after total colectomy, you could see that there are individual little dots. You can see these are polyps. So these individual polyps were taken out, and then they were grouped whether this is polyp is benign, dysplastic, or adenocarcinoma. And we also have just normal mucosa sample, and then we also have blood as a germline. And then we did a lot of omics on these, whether it's transcriptomics, genomics, metabolomics, and several other epigenomics and some spatial stuff as well. So this is the biobank that was led by Christina, Mike Schneider, and William Greenleaf at Stanford to create this large biobank of FAP patients across different modalities. And we did a lot of multi-omics and also the genomics and then epigenomics and so on. So today I'm going to talk about the genomic part only, that I call it. And I've been also part of the others as well. So the whole collection, of course, that paper came out from the Ashton Consortium was the special issue in Nature. If you are interested, you can read more. And there is a website also. The data is available for the community to use for research. So for the on the whole genome side or whole exome side, we use a cohort of 6 patients, and we had hundreds, around over 100 polyps from these 6 patients, and these also at different stages. So you could see that there are mucosa, benign, dysplastic, and so on. And then we also use 2 independent cohorts for validation. One is from Beijing, Peking University Hospital, and then the other one is from the UK. So the one from Peking is also an FAP, but the one from the UK is a sporadic CRC cohort. And what we did was we reprocessed everything again for these other public cohorts, I mean these 2 cohorts as well, using the same exact pipelines. And the only difference between our FAP and the other FAP cohort is that we take 1 polyp and then sequence. So this is 1 polyp, 1 sequence. But the FAP cohort from Peking University is that they take 1 polyp and then they do multi-region. So they take multi-regions from the same polyp and then they sequence. So it's like a multi-region sequencing approach. So it's likely you're going to see a lot of sharing in that cohort compared to our cohort. So you could see that this is just a typical so-called onco plot that we say, where you could see these are CRC driver genes on the left side, and then each column is a polyp, and they are ordered from early stage to late stage. And then of course the color is the mutation type or copy number type. So you could see that there is a lot of APCs happening. So this is the first row is an APC. This is a somatic APC, not germlines. All of these, most of these, I mean, only one patient doesn't have APC, the A014, but rest all have the germline APC. And you could see this is the second hit of APC, and also we have KRAS. This is pretty much known in the CRC settings. And then on the other side, you could see these 2 other cohorts. We see somewhat similar patterns in terms of the mutations of different genes, which are known to be mutated in CRC settings. The only difference you can see that there is a lot of sharing within one specific patient in the cohort from Beijing, just because, again, I said this is a multi-region sequence, so you expect more sharing compared to this is like multi-polyp cohort. Initially, we looked at some quick at exonic mutation burden, and we don't see much of a difference between benign versus dysplastic in terms of the mutational burden at the exonic level, and so does in the other 2 cohorts. We look at the copy number. There is some increase in terms of the copy number burden from early to later on. And then we expose this one, for example, here you could see that on the TCGA setting, the mutation burden, and you could see that the sporadic CRC cohort is way ahead in terms of the mutation burden. That makes sense. And this is a sporadic CRC, and there's a lot of polyps they are in later stage, and then the other, you could see that the PKU cohort also it's later stage because there was a lot of dysplastic samples there compared to the other ones. Okay, so looking at the driver, alterations in terms of at the APC and KRAS, you could see there's a lot of driver alteration in APC and KRAS were common in polyps. 83% of benign polyps, and also 82% of dysplastic polyp had second hit of APC driver mutations or LOH, while only 70% of the normal mucosa samples had biallelic APC inactivation. Further, we see that 17% of benign and 35% of dysplastic polyps had both APC and KRAS somatic alterations. And then again, you could see this is just looking at the multi-hit. Majority of the dysplastic polyps have more than 2 hits of the APC, and also similar for a lot of dysplastic has either 1 or 2 hits of KRAS. Okay, so many polyp samples, you could see that in our cohort lacked clonal drivers mutation. This observation is not consistent with the hypothesis that APC second hit mutation or other driver mutations are required for monoclonal expansion, leading to polyp formation. Furthermore, 8 of 96 polyp harbor more than 1 APC somatic and driver mutation. So with this, we thought whether this whether these clones, whether they are whether mono or polyclonal. So we try to investigate that. So for example, if classically cancer is assumed to be monoclonal, meaning that a single cell acquires a driver mutation like an APC and expands to form tumor. However, detecting this initiation even in human is increasingly difficult because by the time a tumor is malignant, clonal sweeps have obscured its history. Previous studies using limited markers hint that early lesions might be diverse, but we lacked systematic genomic resolution to prove it until now. So, for example, you could see that by if a polyp is polyclonal, you would you could expect that these going at the crypt level. So these are, you could see these are single crypts. So crypt are, I would say, the building block of a colon in the inner lining of the colon formation. If there are multiple distinct mutations happening at the crypt level, and then those expand to form this lesion, and you could see that there is a lot of there is less, I would say, sharing. There's a very distinct things happening at each level, and then you could see that this is like a polyclonal. And if it is just one single crypt sort of expands and forms these polyps, and then you could see there's a lot of sharing happening in a monoclonal setting, right? So then, of course, we use sort of these MRCA, most recent common ancestor, to detect it. How do we detect if a polyp is polyclonal? We use the somatic mutation as a molecular clock by quantifying the number of clonal mutations, those present in every cell of lesion. We can estimate the age of MRC. If the MRCA is existed after the polyp form, the lesion is monoclonal. However, if the MRCA dates back to infancy long before the polyp appeared, it implies the lesion consists of lineages that diverged early in life and came together to form the polyp. So this is our sort of definition, of a polyclonal lesion. Okay, so you could see that by monoclonal lesion, you could see that the how to say more clonal mutations, and so means that more sharing. So you have these mutations happening early on before the MRCA, and then you could see there's a lot of sharing. But in polyclonal, you could see those mutations happening after MRCA, and you could see there are fewer sharing, but these are very distinct mutations happening at individual trunks in the tree, right? By just using the MRC as a cutoff, we found that 40% of benign polyps were classified as polyclonal, and 20% of the dysplastic polyps, were polyclonal. So this suggests that the lesion formation is not strictly single cell event, right? And this is again just using the other cohorts as well. But how we can prove it? I think we have very powerful data set to prove that. So what we did was we did single cell crypt level, sequencing, like single cell crypt isolation followed by whole genome sequencing. And initially we did isolated crypt from a polyp, which is not tumor yet. It's just a normal polyp. These crypts for 20 we were able to isolate. And then on the other side, polyp which is already cancerous, he isolated 10 different crypts. These are individual sort of building blocks of inner linings of the column. And you could see these are the microscopic level images of these crypts. So here I'm showing you a polyp for 20 different crypts. And at the bottom, you have just a normal thing. You could see that is clustering differently. And we don't see any sharing among these 20 crypts. You could see that there are distinct APC mutations happening at individual trunk. You could see an individual clone, and that's suggests that this is a highly polyclonal. These individual clones are taking shape. And on the other side, with the Apollo, which is already cancerous, but we see a different story. So there's a lot of shared mutations among these 10 crypts. You can see there is one event happening very early on. This is the APC event happening, and then of course that event is just taking share across all these, and there's some independence of clones for sure, but that is APC event is in all these clones, and this shows clear monoclonal expansion. So what this suggests is that cancer starts as a polyclonal, and then there is a transition phase happen where one clone takes over, like clone sweep, and then monoclonal expansion is happening. So this was beautifully shown in an independent study that was parallelly submitted to Nature together, where they engineered mice to get FAP. And you could see that in earliest lesions, there is a lot of polyclonal origin, but in the later stage becomes, pre-invasive or neoplasia. And then when it becomes tumor, you can see that just one clone is taking over and a clonal sweep happens. And so there is this, polyclonal to monoclonal transition happening at, of course, these. So I would say that tumorigenesis is a multi-step, like for example, two-phase process in this setting. So initially cooperation happened like at the polyclonal level, and then followed by competition. One clone takes over. Of course, what happens to other clones? Maybe they are lingering around. We are not able to detect with the technology we have. And then there are several other questions. So the idea, of course, so here is the sort of the summary. In summary, so this study demonstrated that tumor initiation is not strictly single cell event. Polyclonal is frequent, for example, in benign 40%, and dysplastic 28% polyps were polyclonal. And malignancy is marked by the transition of molecularity, and future work, of course, must focus on cell-cell interactions and also microenvironment signal that allow these diverse lineages to cooperate in earliest stages of the cancer initiation. And of course, the question of mono versus poly, it's not new. The debate been quite long, so there's quite a lot of evidence that suggests polyclonal origins in various cancer types. So if you're more interested, of course, I would redirect you to this review by Parsons from 2018 to learn more about this debate. So all of these data sets and results that came out, They are available for the community use in terms of the code and the summary data sets. So this is the people that I would like to thank from Stanford that were very helpful driving this last part of the study, and also the first part of the study. So also I have a lot of collaboration that happening right now. A lot of studies that perhaps will present in some of the future seminars. A lot of collaborations happening within the country, in the region, and also internationally. So it's my group, and I'm happy to take any questions. Thank you.
Thank you, Dr. Aziz. We have a few questions. Maybe you can take them. So one of the questions, it's very generic. This is from Farhan Ikhtiyar, and he's asking, can you explain in simple terms how AI helps doctors understand a person's DNA faster and make better treatment decisions?
Understand. Okay. So of course, I mean, DNA is just the sequence, right? And we don't know a lot of functions for sure for a lot of the DNA. So for example, we only know, for example, the protein coding genome, which is just 2% of the DNA. Majority of the DNA we so-called, been calling it sort of the junk DNA, which is the non-coding. We still don't know a lot about the function of this DNA. So AI can help us to decipher these regulatory, I would say this non-coding genome plays a lot of regulatory mechanisms in terms of when to turn on a gene and how much to turn on and how much to turn off at a specific time point, in whether in development or disease onset or and progression. And AI can help us to understand this the regulatory grammar of the so-called the junk DNA by bringing a lot of different modalities in by modeling. And I think that's the big, I would say, benefit of AI to integrate these multimodalities of data set to understand the non-coding genome. And then once we know that, of course, this non-coding genome or the regulatory grammar could be used in terms of early detection or maybe in treatment, targeted treatment by targeting maybe a specific part of the DNA, which plays an important role in whether in tumor initiation or disease initiation and so on.
Yeah. So we have another question from Dr. Al-Hawajri. How can AI in healthcare be used to specifically detect early signs of patient deterioration in critical care units and predict potential adverse clinical outcomes?
Right. Again, this is the same if you have, for example, enough evidence by integrating a lot of data sets, and whether it is imaging, whether it's genomics, and can we combine those to understand, what, of course, disease onset means and what in response to specific treatment, and also, the path in terms of the clinical care, how that goes. Of course, once we sort of connect these medical health records and imaging and genomics together to build these holistic models, and these are more, I would say, also not just built based on European data sets, but also maybe built on local data sets, because genomics is very diverse. And then of course we will be able to use these AI health models in the healthcare setting to understand individual patients using their, I would say, longitudinal data set, if we have access to those data sets. But of course, I think a lot of, I would say, openness need to be from the medical community, from the hospitals, and of course there is a lot of data privacy and so on. For example, UAE is building the Emirati Genome Program, which is it's an amazing treasure trove to understand the whole population, because most of the population being sequenced and then of course they have access to the medical health records. Can we combine those to understand and then use this as a diagnostic way? It's I think there's a lot of amazing opportunities.
Great. There's a couple more questions actually. Do you think foundation models will become important in genomics the same way they became important in language and medical imaging?
Yes, I think that they are coming, I would say, to at least a single cell, understanding transcriptomics. It's becoming more and more. And then, of course, the non-coding genome also there's a lot of unknowns, but still understanding the non-coding part and then building these foundation models for non-coding DNA is also getting more and more. I think there is a lot of, this is also happening, these so-called virtual cells and so on, and there's a lot happening on that front as well. So I think there is a lot of room to improve, and they're gonna be, I would say, more and more, definitely. Yeah.
Great.
But in
There's
terms
one
of
more question. Oh, sorry.
the correctness or the validation, and then I think we are still, I would say, early on to tell. Of course, the AI, I think, simply predicts the next word, right? But people are doing now, of course, predicting this next, medical event. There's a lot of things happening there as well in terms of these foundation models.
Great. There's a couple more questions. Considering how far AI has come already, would it be able to reliably predict molecular features, treatment response, etc., directly from histopath slides? So reducing the need for additional molecular profiling?
Sure. I think there are people trying to predict at certain level using just histology images, and there is a lot of foundation models for that as well. I think there has been a lot of progress that's been shown, at least at the basic research level, but I don't see yet anything went into the clinical setting yet, but I think it's coming.
Great. Another question, is there an AI genomic study on cancer prostate for early discovery and management?
Definitely, yeah. I'm sure there are, at least for early detection. For manage, I'm not sure, but for early detection, there is a lot of AI-based studies on prostate cancer as well.
Great. Just one last question for myself. This is all like tools and data for multi-omic and cancer genomics, but is there a similar thing for like rare disease, Mendelian genetics models for this field?
Definitely, I think there are people are working on those as well, but the data set is, I think it's small for rare diseases to do, I would say, predictive modeling and stuff. Of course, if if they're able to get enough data points or samples, definitely there is room.
Great. Thank you so much, Professor Aziz. We'll move on to the next talk. Professor Yaqub.
Thank you.
Salam.
Let's share sound. Can you confirm that you can see my screen, please?
Not yet. Yes, we can see it.
Okay. Shall I start now? I have 45 minutes, right?
Yes, please go ahead.
Okay, so thank you very much for having me today to give you a talk about AI in healthcare and to focus on really some of the research outcomes from Mohammed bin Zayed University of AI in the healthcare domain. And before me, my colleagues from the university showcase some of the great work that they've done. And my goal today is to really focus on how was AI in healthcare, how was it in the past, and then focus on the current present, and then also what would the future look like for AI in healthcare. So let me get started. Before I do that, just a bit of introduction about myself. I'm an associate professor in the computer vision department. I've been working in AI in healthcare for almost 19 years now. And I am also a founder of multiple companies. Prior to coming to the UAE almost 6 years ago, I did my PhD at the University of Oxford, and then I worked almost 10 years at the university as a researcher. And this is my team, who are like a set of amazing PhD, master, and postdocs who work tirelessly in many different problems in AI in healthcare. And today I'm going to showcase some of their work in this domain. I do collaborate with many entities in the UAE, including many hospitals and regulators, and outside the UAE as well. In a nutshell, if I will summarize my work later on, but in a nutshell, I work mostly in medical imaging, but I do a little bit of work on non-medical imaging in different domains, including oncology, neurodegenerative diseases, fetal health, and cardiovascular diseases. And I'll mention these a bit later on. I'll go in a bit more details. But before I talk about my research, let's just look at the AI in general. So just also explain some terminology that they might be not very clear to everyone. I know everyone is saying AI, but honestly, I found that many people don't know what AI exactly means. But in very simple terms, we know it just refers to the fact or the tools and the algorithm that allows us to make the computer smart. But the real thing that we have in the current revolution of AI is not coming from the AI as a whole field, but rather from a subset of it, which is deep learning. In AI, we have a subset of that we call machine learning, which is a set of algorithms that allows us to learn from experience. Take an example. If you are a first-year medical student, you go to the university, the doctor or the professor in the university will show you how to distinguish, say, pneumonia from a normal patient by showing you maybe X-ray images and telling you, okay, look here, you see some patterns, and blah, blah, blah. This is called supervised learning, which is part of machine learning. And when we teach the computer to do the same, we do what we call machine learning. These are algorithms that learns from data. But the real revolution of AI is it started from the reinvention of deep learning. And in simple words, it's a subset of algorithm within machine learning that allows us to learn from, apologies, that learns through a method that mimics a human brain's learning by creating a set of artificial neural network that are able to learn from the data in a way mimicking our brain. But AI doesn't stop here. I mean, there are many other terminologies that you probably have heard of, like computer vision, robotics, NLP. And if we really think of the last 3 to 4 years, we live in the era of what we call generative AI, not the era of standard AI. So we've been doing standard AI for many years, which simply refers to the AI where we take data in, or the machine learning that takes data in and gets outcome or decision out. But now we are in the revolution of generative AI, which many of us use, such as ChatGPT, Gemini, Claude, and many other tools that allows us to put data in and get content out. Like you can create an image from prompt. You can generate a video from prompt. You can summarize an email. You can draft a response to an email. And that's what we call generative AI. The reason I want to give you this introduction is just to give some background on what AI is, so I can move on to my past, present, and future of AI, especially in healthcare. So if I want to summarize this revolution of machine learning, I could probably focus on 3 main revolutions of machine learning. So ML is for machine learning. So the first one is actually prior to what we call deep learning. So that's, you can call it from the start of machine learning, which is almost 70 years ago, to almost 2011, 2012. So that revolution, what we had is simply we have some problems that we want to solve with machine learning. Like take an example, I want to have a method that looks at the electronic health record of a patient and assess or predict whether the patient will have sepsis or not. Okay. So in that initial revolution, which we used classical machine learning, we used to do that era, we used to call it as well feature engineering era, which means we used to craft our features. In other words, if I'm trying to distinguish brain cancer MRI scan from non-cancer MRI scan, is that I had to tell the algorithm what features it should look at in the data to distinguish that. For instance, for glioma, you should tell it this glioma looks like this. It has this bright region that appears on this T1, MRI T1, and so on and so forth. While normal MRI should have a two hemisphere where they have to have a bit of symmetry, blah, 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 and so on. So you should program your features. You should, what we call feature engineer, or engineer these features. And this is actually one of the reasons that we couldn't at that time make AI work really well on data until we moved to the second revolution. In my opinion, we call it the deep learning revolution, where what we've tried to do is to, instead of telling the computer what features to look at, we build algorithms that can sort of exhaustively try to figure out the best combination of features from the data. So more of purely data-driven. So instead of me giving it MRI scans of glioblastoma patient and normal patient to distinguish brain cancer, I would rather just give it the data without telling it what to look at, invest in curating the data, in getting that the data is analyzed by doctors. And the doctor told us that this MRI has a cancer, this MRI doesn't have cancer. And deep learning models doesn't require feature engineering, which means they go and zoom into the data as much as possible to figure out the set of features that are best distinguish the two categories in this example. And this is based on what we call artificial neural network and a version of it called convolutional neural networks. Okay, so that's the second era that we've been through. It started almost roughly 2012. We've started to see the impact of it. And then gradually we moved into the third revolution, which is what we call the revolution of foundation models. And some people may know it as, really, maybe have seen it from the start of ChatGPT. There's a difference between ChatGPT or foundation model, but let me just maybe link it to the creation of large language models. So some of us, maybe when they're sick, when they have a problem, they might just go to ChatGPT or Gemini or Claude and say, I recently started to have headache and I have this and that, and you give all the symptoms, okay? And you try to ask ChatGPT to tell you what's going on, which is one way. I mean, some people have done that. So the question is, how does this model understand health? Now, many of them do understand health because they're more of like a foundation model for a problem. The concept of foundation model is more or less means that you build an AI model that is able to understand many things around us and able to answer question even though maybe we did not train it on that question. Some people think of ChatGPT as a foundation model. I would rather think of it as a large language model, but more a foundation model is usually more specialized than these generic LLMs. So an example of foundation model that's been trained, for instance, Google has launched a foundation model for medical data called MedJama, which understand medical problems, which is likely to be more accurate than generic large language models. The goal we wanted that is not to create most of the AI before the Revolution 3 is specialized, very, very specialized. So you build a model that just look into, I don't know, acute kidney injury estimation or prediction, or sepsis prediction, or length of stay in a hospital. These are very specialized models. The goal of foundation model is to build AI models that are a bit more generalizable, that can work across the board in a wider range of applications. Okay, but I want you to understand that this hasn't been like just disjoint revolution. It has been a continuous revolution. So things has moved from one step to another to another. We still until now sometimes use classical machine learning to create predictive models depending on the problem. Okay, so that's the foundation of what I want to build on. So from now on, I want to move on and tell you a little bit more into some research project that my team and I are working on in the UAE and some outcomes that we have. So I work across the board from early age, as early as fetal health, during pregnancy all the way to later age to neurodegenerative diseases. So my team and I work on fetal health. This is a very active domain that we work on in collaboration with different hospitals, where we assess fetal growth. We build AI model that can tell whether the fetus has, I don't know, congenital heart disease or not. And a good news we had last week, I got one paper accepted in Nature Partner Journals in Digital Medicine on showcasing how our AI models developed here in collaboration with Cornish Hospital can predict congenital heart diseases with sensitivity exceeding 80%, where doctors is about 50 to 60%. So that's one set of theme of projects that we work on, but we also work on neonates and kids, so in pediatrics, where we assess different issues. One of the problems that we worked on with coordination SKMC is two major problems. One, how could you predict kids who are likely to be born prematurely, but then follow up with them if they are born prematurely, what type of disease they might have. When a kid is born premature, one of the major problems they have is that their lungs are one of the last organs to be developed, which may lead to like inability to breathe well, so they get incubated. But then they might suffer from different problems, including non-respiratory distress syndrome, neonatal respiratory distress syndrome, or transient version of that, or maybe pneumonia or other things. So what we did is we built AI model that can diagnose kids when they are in the neonatal unit. In pediatric, we did different projects, including one of the interesting projects is in leukemia, and the research question was on to do leukemia diagnosis or subtype diagnosis to a different type of leukemia, including ALL, AML, CLL, CML, whether it's acute or chronic, or what type of acute or what type of chronic. The way to do it is you have to do bone marrow biopsy, which is invasive, time-consuming, very expensive. And we do that to put that under the microscope, then you diagnose leukemia. So in collaboration with different hospitals in the UAE, we build AI tools that's able to diagnose leukemia from a microscopic image of a blood sample, rather than doing bone marrow biopsy. Moving on, in cardiovascular diseases, I'm not going to mention much here because I have a whole slide for it later on. So we work across the board from echocardiography to CT angiography and others. My team and I also work on cancer. So we have work on different problems in cancer, including head and neck cancer, colon cancer, and other type, or brain cancer, and other type of cancers. And finally, as I said, my work is across the board from early age to later age. My team and I work actively on neurodegenerative diseases with a big focus on dementia, and specifically for Alzheimer's. In addition to that, my team and I work on other problems, including maternal health, acute kidney injury, antimicrobial resistance, pulmonary diseases, autism spectrum disorders, and others. So what I'm going to do in the coming 4 or 5 slides is I'm going to pick few projects and zoom a little bit more and show you some outcomes that we developed in this research in here. So let's move on to the leading cause of death worldwide. So if you don't know, the leading cause of death worldwide is cardiovascular diseases. And I think the stats I have is an old stats, but in 2020 maybe, or 2019 actually, the number of people dying from coronary artery disease is approximately 19 million people, according to WHO. That's just the number of people dying. There's a huge number of people who get affected. The question that my team and I focuses on, the research question, how could AI help diagnose patient better, but more importantly, predict the future, tell which patient might have a problem as early as possible? So this is the research question that we're trying to solve. So under that, I'm going to show you 3 studies that we worked on. So the first one is in collaboration with multiple hospitals and also the University of Oxford. And it's under a big study called the Orphan study, where we try to come up with AI-driven novel biomarkers to predict stenosis in the heart, in the arteries before it happens. So usually a patient comes in with a heart attack, is like already a blockage. The plaque is already there. There's a full stenosis or major stenosis inside one of the arteries somewhere. So it's usually too late, right? The question we really want to answer here is, could you come up with a way to assess stenosis before it happens? And it turns out you can. And the way we did it is that we found novel biomarker. We call it fat attenuation index. That allows us to measure some markers on the fat that surrounds the arteries. And if you can quantify inflammation, because before stenosis happen, the fat around the artery where the stenosis will start to appear, that fat becomes inflamed. You cannot see it by eye, but you can quantify it by computer or AI. And this is looking at CT angiography. Another project that we're currently working on is building foundation models for echocardiography that can automatically help compute many different biomarkers or markers from the echocardiography scan. You know, CT angiography has radiation, ultrasound is cheaper, no radiation, but it's very subjective and really the quality is not super good compared to CT. But as I mentioned, it's radiation free, it's cheap, and it's easy to get to the patient compared to the other one. And hence we were working actively on echocardiography. So recently we've developed some work that is able to predict ejection fraction, systolic diastolic dysfunction, aortic stenosis, and other markers automatically through AI. We call this system EchoVision. And to make it even more affordable and accessible to people, we also focused on ECG and how could we really do things with AI and ECG that usually cardiologists do not see in ECG. So for instance, you cannot, as a cardiologist, assess systolic and diastolic dysfunction properly from an ECG. You can tell other things, you can look at arrhythmia, you can look at other problems, but what the research question we want to answer in this study, which is in collaboration with a big hospital in Taiwan, is that can you predict systolic and diastolic dysfunction from ECG? And it turns out, according to the table on the left-hand side of this slide, that with a high degree of accuracy, I mean, it's compared to normal way, is that you can, with an AUC exceeding 80%, you can predict systolic dysfunction, and diastolic dysfunction is harder, but you can do that with an AUC of 0.7. We looked into other things, honestly. We looked into diabetes, we looked into PE, whether ECG can tell us more information about the patient. Moving on to another problem, which is what we call report generation. We know the largest field, if you look at the largest number of FDA-approved AI in healthcare products, the majority of them are in radiology for different reasons. So last number I looked at the beginning of 2026, when I looked at the number of tools in FDA, 77% were in radiology. Okay. So one of the main important things in radiology, I'm sure my colleague Hisham might have talked about that, is to do report generation. So the question is, how could you do report generation? So a scan comes in, a report is spit out, and the radiology, instead of writing from scratch and reviewing, can actually just review, improve, and then just apply, and then just accept it. So here's an example. So some of you might be clinicians. This is a scan of a patient who has a problem. So I'll leave it for another 10 seconds to think of what could be the problem. So when you pass this one to an AI tool to tell you what's going on, it just simply writes your findings, impression, and differential diagnosis. And it turns out that this patient has macroadenoma, which is a type of cancer. And when I reviewed it with some doctors, it was correct. And I'll show you another case of automatic report generation that we did with SSMC. Here's an example that we did for CT report generation, where it doesn't only write the report, but also generate images. So it allows you to see how the image, or generate synthetic chest X-ray images, like this one on the right is fully synthetic, generated from just drawing a mask. And at the time you were able to put a pathology on it. So for instance, put say cardiomegaly or pneumonia or other things. So say put pneumonia in this region and then generates the chest X-ray on that location. At the same time, it will allow you to create reports. I have a recent paper just published last week on echocardiography report generation. So you get an echo scan and you'll get the full echo report automatically generated. Moving on to another topic, which is close to my heart, is fetal and maternal health, especially fetal. This is where my team and I work actively. It's been working on this one for about 17, 18 years, honestly. This specific study is with Cornish Hospital, where we have point of care ultrasound. These are small devices. And the idea here is that if you can diskill people, you don't have clinicians all over the world. Like the number of obstetricians in some countries is probably, you can count it in your hands. Okay, so we have a very huge shortage of doctors all over the world, especially in developing countries. And this study is looking into how could we build AI tools that can you put this transducer into the hand of, say, a nurse or a midwife, and she scan a pregnant lady, and then the AI tool is running on her smartphone to assess fetal growth. In this example, you see something in green. That's the abdomen of the fetus, gets assessed by the AI to measure the abdominal circumference, which is a biomarker or marker for fetal growth. It's just an example. Another interesting study that I work in cancer, but let me focus on head and neck cancer. So we lead a large-scale study in collaboration with 11 hospitals. It's a part of an international challenge for head and neck cancer, how we quantify head and neck cancer and stage head and neck cancer and prognose the future outcomes. So this is part of a major study, as I mentioned, 11 centers from many different countries. We now collaborate with SSMC, Burjeel, and Cleveland Clinic on similar study, where we developed AI method that can take the PET and CT scan and allows you to create, diagnose the patient, quantify tumor and lymph nodes, stage the cancer, and probably more important thing is to tell you the future outcome prognosis, tell you what's the likelihood of recall, what's the likelihood of death, what's the likelihood of other things. Okay, so all this has been published and you can see it and you can find it online. And maybe the last one or two studies I want to mention is in neurodegenerative diseases. Before I mention neuropath, let me also just pause here on talking about multiple sclerosis. I don't have a slide for that. We've been actively working on multiple sclerosis. We build AI models that can detect new lesions. So in MS, the problem is that the patient comes in at multiple times and then they'll have a baseline MRI and then follow-up MRIs. And then one of the major questions as a neurologist and neuroradiologist, you need to say whether there are new lesions or not, and or how the current lesions change over time. And so what we did is build a model that's able to accurately segment new lesions in follow-up MRI. And then the other thing that we did in MS, which haven't published it yet, this is we built think of it like a ChatGPT for MS, as an LLM that is that's been trained on a large number of research papers of MS patients or papers and MS information or patient data that's able to answer questions and help support doctors, patients, family, and community answer questions properly about MS. So that's MS, but let me now come back to this slide, neuropath. This is part of a study that my PhD student Selma and I have been working on it for almost 3 years now, where we wanted to, our goal was 3 things. We wanted to early detect preclinical Alzheimer. So how could you tell that this patient may have Alzheimer as early as possible? The second question, how can we help diagnose the type of dementia? You know, dementia is an umbrella of diseases, including Alzheimer, vascular dementia, MCI, and the patient could be normal as well or preclinical. So the second question, research question we wanted to answer is how could we do differential diagnosis of different type of problems, conditions.
Dementia diagnosis.
And the third question was about how could we help doctors assess the brain over time? So longitudinal analysis of the brain to assess brain atrophy over time. So that's what we did. And then recently we built NeuroPath as a tool. That is, you can access online on this website. And now we're testing it at different hospitals to assess its efficacy and safety. Shortly, I'm going to show you a video about NeuroPath. So I hope, please, the organizers, if you don't hear the voice, let me know, stop me. But I shared the sound, so you should hear the voice. Let me start it.
Dementia diagnosis is inefficient, expensive, slow, and inaccessible. NeuroPath changes that. Clinical grade dementia detection, reimagined. 1 MRI scan, 1 automated pipeline. 55 million people currently live with dementia worldwide. 65% never officially diagnosed. 1 new case every 3 seconds. NeuroPath was built to close this gap. Powered by a spatiotemporal graph neural network trained on 10,000 annotated cases. The upload interface supports both single scan and longitudinal tracking modes, with NIFTY and DICOM support. File metadata is automatically verified. The quality control panel previews each scan in all 3 anatomical planes before the pipeline runs. Every brain structure is color-coded and overlaid on the MRI at adjustable opacity, fully navigable across all slices, sagittal, coronal, multi-view, every anatomical plane available. In multi-view mode, all 3 planes render simultaneously with synchronized crosshairs tracking the same anatomical coordinate across all panels. The 3D view renders the full brain interactively. Every structure is individually toggleable. Deselecting the cortex exposes the full subcortical architecture in 3D. Hemisphere asymmetries are calculated automatically. Selecting 2 time points activates the side-by-side comparison view, all planes synchronized in real time. In 3D, the progression is immediately visible. The longitudinal table quantifies every change, every structure, every time point, automatically tracked. The radiomics dashboard, 3,424 features extracted, outliers flagged relative to the normative cohort, structure ranking and feature importance are displayed. Every feature is benchmarked with a z-score, percentile, and status flag. The analysis is also available in longitudinal mode. The comparison tab of the volumetric analysis tracks feature-level change between time points. Each row shows the time point value, absolute delta, percent change, and a categorical trend status. The classification results page. Switching between time points shows how the contributing structures shift as disease progresses. Clicking View Highlighted Regions takes you directly to the segmentation viewer, where the top contributing structures are highlighted, turning the classification explanation into a spatial, anatomical walkthrough of the decision. The final output, the NeuroPath Analysis Report. A single export action generates the complete clinical deliverable. The analysis summary integrates classification results, key contributing features, from a raw MRI scan to this complete, structured clinical report. That is NeuroPath.
Okay, so with that, that's all from the research I want to mention. But before I give you the take-home message, I want to also talk about the future. So when it comes to AI in healthcare, healthcare is a very rigid business, honestly. I've been in it for a very long time. In fact, when I came into Abu Dhabi 6 years ago and started as a researcher and professor at the university, I'm always like thinking of business. Now, I thought I'm not going to open a company in healthcare. Simple as that. When I open a company, it's going to be something else, because I've been through it in the UK, and it took ages to get to FDA approval. So the last product I did in FDA approval took 6 years. So it took 6 years from the time that I started working on it. Actually, a bit more than that, but let's say 6 years to get it FDA approved and functioning on people. And I'm proud that that system I was talking about, which is related to fetal health, it's now used on millions of women all over the world for anomaly screening. But what I want to mention about the future is that, honestly, the healthcare domain is probably the domain that is the far away from other domains when it comes to AI adoption. There are AI adoption in different hospitals, and especially advanced ones, but it's still limited. So the future I see is that putting proper regulation is one major factor in getting AI to be used more in healthcare. So there are definitely, again, there are many different systems that's used, but still this is just touching the surface. There are some hurdles we have to go around, including regulation and including integration, because one of the major problems that many hospitals, they have legacy system that may not be easily used with AI technologies. And I've suffered that myself with trying to put some of my tools in hospitals where it wasn't easy. Not just because of the integration, but also the regulation and the rules by securities, teams, and so on. So the way I see the future is that we should hopefully start to see now more and more integration of AI technologies in healthcare compared to other businesses where we see it much more often. Okay, so as a take-home message, just as a revolution in AI in general, we started from rule-based system. That's the old days in AI. Then moved to machine learning, then to deep learning, and now we live in the era of reasoning or LLMs and foundation model. And honestly, maybe I should not say that, but the problem is mostly not in the algorithm. The algorithms are really important. This is something that I build. But honestly, the problem is usually in the data, the quality of the data, and the workflow and the regulation around it. The future should be proactive, not reactive. What I mean by that, honestly, something I keep saying because I learned that from a friend of mine. We say healthcare, that's wrong. See, in fact, my title here in the left-hand side is called AI in healthcare, right? That is wrong. It should be AI in sick care, because what we have now is sick care rather than healthcare. We don't do healthcare. What sick care is that someone gets sick and then just try to get care for it. That's what we have. To get healthcare, we have to be proactive, which means we have to be getting help rather than sick, then be treated. And that's what we should be thinking about. How could AI help us be proactive? And at the end, it's very important that people ask me about this. Oh, this will replace me and this will take my job. No. Yes, AI will replace some jobs, but not necessarily in healthcare. AI, simply we keep saying that it will replace clinician. It will not replace clinician. It will replace. So AI will never replace a clinician, but rather the clinician who uses AI is likely to replace a clinician who doesn't. In other words, if I have a hospital and I have a position for, I don't know, urology, and a urologist who's using AI tools to do his or her task much better, I would rather hire that person than hiring a person who's not doing that. I'm a little bit earlier in time, but I'm more than happy to take questions. So these are my references that most of the work is presented today is some work, already published work from my research. So please have a look into my research on my group website, which I'll show you now. And with that, thank you very much for your time, and I'm happy to take questions. And this is my email address.
Thank you so much, Professor Yaqub. There are many questions, a couple of questions in the chat in the Q&A. So someone's asking, for someone entering medical imaging AI today, is it still worth investing time in learning radiomics and traditional radiomics-based machine learning pipelines? Or would you recommend focusing directly on deep learning frameworks like MONAI?
That's a very good question. You could go and really learn MONAI, definitely. But honestly, without knowing the existing original knowledge from machine learning and radiomics and bioradiomics and so on, you will reach a limit where you cannot put good understanding into future things that you will learn. So my suggestion is learn both of them. You might want to start with this one that you're more comfortable with. So if it's me, I would start with radiomics, with classical machine learning, and gradually move to MONAI and other tools. Because honestly, AI always reinvents itself. Believe it or not, deep learning is nothing new. I said the revolution happened in 2012. But in reality, the algorithms, the original algorithm, they were there at least 10 to 20 years before that. Okay, but we couldn't make them work. And in 2012, 3 things appeared, including these algorithms have been reinvented in a way that they become more efficient, but also we have more computational resources with GPUs and more data. And that make it a big difference. The point I'm saying here is that what you learn in the old, like classical machine learning, gets reinvented or reinvent itself to be used in future AI algorithms. So definitely invest in both.
Great. There's another question here by someone who has already built an AI healthcare system model in the Republic of Korea, and they pitched it through a government-supported platform. And it works, but the hospitals don't fully trust it, and the regulators have not allowed clinical deployment. So the question is, after building the model, what should be the next step to make it clinically trusted and regulator ready?
This is a tricky thing, honestly. So yes, so the main thing is that you really want to find ways to do clinical trials where you can deploy it, definitely free, usually free of charge. You put it in hospitals, you put it in clinics or units where it gets evaluated through a well-defined process. And to be able to use it in future as a business, you need to have it regulated. For instance, if you go to USA, you have to go through FDA. And FDA will ask you for a few documents to show efficacy and safety. The same thing if you come to the UAE, we have our regulators in different Emirates. So we have DOH in Abu Dhabi, for instance. So you have to go through them to show the regulation. But to reach to that point, you really need to go through a process of clinical trials and clinical assessment to showcase. Many of the AI tools gets work. I mean, usually you do it with hospitals and universities, so you can publish some results as clinical journals supporting the fact that this AI tool actually works. So people will trust it if they. So, for instance, when smartphone initially were created, many people rejected and they just kept using their normal phones until they saw the value. They saw the value that if I use a smartphone, I can use this app, which means that I don't need to go to the bank, but rather open an app of the bank, and then I can do a transfer through that. What I'm trying to say is trusting the AI solution doesn't come in one day. It comes as a process. It comes as publication of research papers supporting the tool. It comes as a clinical evaluation. It comes as a word of mouth. So usually you should find your AI champion from clinicians. So usually when I go and want to deploy something, I find a clinician who's a champion that helps me encourage people to use it, to try it. And then you go through this process all the way to get it regulated in the country of interest.
Great. There's another question here, which is asking, do you think if LLMs are trained on thousands of cancer patients, for example, like their weekly blood work scans, health issues, and progress from diagnosis, prognosis, so can it be used to train a model to predict how someone's cancer is going to progress in the future, just like in video games, basically?
Yeah, so yes and no. So yes, mostly yes, because now we have what we call in AI world models, okay? Something called world models. This is just a state-of-the-art research in the domain. That's why many people didn't hear about it, still in papers, which is AI tool that is able to look into all data and in a way that it can simulate the future. And there are a handful of publications on this, not many, which try to simulate how would this patient cancer becomes if this intervention happened, if this drug is given, if let's say for instance TMZ is giving to this patient, what would be the cancer? So it generates new data that shows you what happens. So yes, we are in this era of having what we call world models. These are models that's able to create the future. But the question is, how good are they? How accurate are they? So at this stage we're in 2026, they're not super accurate. They're more in some domains, for instance, predicting the future out, whether the cancer will come back or not. It's in the range between 60 to 70% accurate. But they're sort of mimicking what doctors do. I mean, if someone, for God forbid, has cancer, doctor will get some information like scans, CT, MRI, whatever, and other information, and say to the patient, you have this cancer, stage 4, you're likely to live 3 months. Okay, that's science. Okay, doctor didn't come up from this number from their brain, just from nothing. They actually saw other patients like this. But the problem is, this is typically inaccurate, and we've seen patients who lived much longer or maybe died much earlier. And that's just because we human can keep in our brain small amount of data to make decision out of it. Although maybe this doctor have seen 1,000 patients in the past, but if we give AI algorithm tons of thousands, maybe millions of cases of cancers, it's able to do much better than us as a human to predict the future. So if someone is interested, read about world models.
Perfect. Okay, there's another one. As clinicians, we often see similar ECGs and echo findings in primary cardiac pathologies and secondary to systemic issues. So how much can current models differentiate between these rather than only identify the abnormality?
I don't have numbers on accurate numbers on in pathology diagnosis, exact pathology diagnosis, but when we did it from echocardiography, not from ECG, we had a range of we had about 20 different findings that we wanted to look at, 20 plus. And the range of sensitivity that we achieved is between roughly 60-ish% to 92%. So I don't have the table on with me now and the name, but for aortic stenosis, we're in the range of 80% AUC, if I remember correctly. So there are already tools that is able to do subtyping or sub-classification for different pathological or cardiovascular findings. From ECG, I'm not aware of many. They're just looking into maybe just mostly arrhythmia and other smaller conditions. In ECG, in echo, there are now tools that can do 20 plus different findings with varying sensitivity.
Great. And just one last question from myself. You mentioned AI in sick care, but what about prevention?
Yeah, so that's the thing. That's what we should be thinking about, actually. How could we prevent things? And you can prevent it by different ways. So you can prevent, I don't know, you can prevent non-alcoholic fatty liver disease, for instance, or later on fibrosis for the liver by changing our lifestyle. Okay, so just some basic intervention can help us prevent things. But also we can prevent by doing screening. So currently screening is not, in many screening, it's not cheap. It's not easy to get. It's not, for instance, for a woman to do breast screening, she has to go to the hospital to do a mammogram and so on. And also there are stigmas sometimes from different countries and so on. But if there is a tool that this lady can just put in her finger and it tells you that your likelihood of getting breast cancer in the coming year is X%. That's easy, accessible, cheap. Okay, the same thing. One problem like I'm doing for screening is that to do screening for fetal ultrasound, you have to get the ultrasound device, put it in the lady, scan her, assess it. What if there is a patch similar to what we put for people who has diabetes? And this patch, you just stick it in your tummy while pregnant, especially if you are a high risk patient, and it just monitors you all over your pregnancy. And this patch is quite cheap, and I'm not talking now, I'm talking in the future, that you can just buy it from off-the-shelf pharmacy. So this is where prevention will be really, really valuable. And we can do that with improved technology and with AI.
Perfect. So with this, I think we should conclude the session. Thank you so much, Professor Yaqub, and to all the speakers. Please, attendees, we're about to launch a poll for you to fill in so that you can get the CME certificate next week, inshallah. Thank you very much, everyone.
Thank you.